First question is, what is the general chapter for UV spectroscopy as per USP? USP General Chapter 857. Next question. What is the principle of UV spectroscopy? The principle is based on Beer-Lambert's law. That is, when a beam of monochromatic light passes through a transparent medium, the rate of decrease in intensity of light is directly proportional to the concentration of the absorbing medium and the path length of the cell. Next question. What is Beer's law? Absorption is directly proportional to the concentration of the medium. More the concentration, more will be the absorption. What is Lambert's law? Lambert's law is absorption is directly proportional to the path length of the cell. The larger the path length size, more will be the absorption. Next question. What is the limitation of Beer-Lambert's law? The limitations are Beer-Lambert's law does not obey at a higher concentration. The light should be monochromatic. The absorption of solution should not be more than 1. Next question. What are the components of UV visible spectrophotometer? There are 5 components of UV visible spectrophotometer: light source, monochromator, sample holder, detector, and recorder. What is the scanning range of UV? The scanning range of UV is 200 nanometer to 400 nanometer. What is visible scanning range? The visible scanning range is 400 nanometer to 800 nanometer. Which cuvette is used in UV scanning range? Quartz cuvette is used in UV scanning range. Next question. Why quartz cuvette used in UV scanning range? Quartz cuvette does not absorb light, that's why it is used in UV scanning range. Which cuvette is used for visible scanning range? Glass cuvette is used for visible scanning range. What is the measuring unit of UV visible spectrophotometer? The measuring unit is absorbance. Next question. What is the light source for the UV region? The light source for the UV region is deuterium lamp. What is the light source for the visible region? Halogen lamp is used as a light source for the visible region. What is the light source for UV visible spectrophotometer? Xenon lamp is used as a light source for UV visible spectrophotometer. What is the use of UV visible spectroscopy? UV visible spectroscopy is used for detecting functional groups. It is used for qualitative and quantitative analysis. It is also used for detecting maxima of the compound. Which compound does not absorb light in UV visible spectrum? The answer is chloral hydrate. What are the calibration parameters for UV visible spectrophotometer? The parameters are control of wavelength, control of absorbance, limit of stray light, resolution power. Thank you.